Hi, my name is Jean MacRae from the Department of Civil and Environmental Engineering, and I'm happy to talk to you today about the contaminants in our food waste and whether it can be safely recycled to circularize the food system. So food waste is a major problem throughout the world. Globally, about a third of food that's produced for human consumption is wasted, and this comes at an enormous cost. Obviously, economically, somebody along the system has bought the food that ultimately is not consumed by humans. But also, there are social costs associated with that, some of it intertwined with the economics, but also there are just flat out a lot of people on the planet who don't have access to uh, enough food. And from an environmental standpoint, there are many, many issues associated with pollution and the effects of wasted food in our waste management systems, the input materials that are not productively used in the end, since the food isn't consumed as it's intended to be. So one of the reasons that the system is so wasteful is that our current food systems are linear. So we have agricultural production, processing and packaging, distribution and retail, ultimately consumption of the food. And at each stage, there are inputs into the system and waste outputs that really, to me, indicate inefficiencies within the system. So the kinds of inputs that I'm talking about are things like water, fuel, fertilizers, um, packaging materials, additional additives that go into the food, and labor. And at any stage along here, there could be food that's wasted, but also a lot of the other input materials may be wasted, or a portion of those materials may also be wasted. So this is inherently an inefficient system, and the individual parts of the system don't sort of talk to each other the way they should. So let's look, I'm just going to look at a couple of the inputs into the system and discuss a bit of how those inputs are problematic. Phosphorus is a major component of fertilizer, and it's a limited resource. We often don't think about the sources of phosphorus, but most of it is mined, and the vast majority of the mineable, inexpensively mineable phosphorus resources on the planet are in Morocco. And that's not in the US, so we don't have control or complete control over it. Morocco is a good ally, but nonetheless, it isn't here. There are some resources in Florida, but there's many environmental issues associated with its extraction. As phosphorus fertilizer becomes more scarce, we have to move to more difficult to obtain types of phosphorus, which is going to increase the cost and potential pollution impacts. Fertilizer production through the nitrogen component, so through the Haber-Bosch process, is highly energy intensive, and just the production of fertilizer globally accounts for a little over a percentage of total global energy use. So reducing our reliance and requirement for fixed nitrogen is an important environmental factor. On the other side, so on the waste side, the question is, why not landfill food waste? Is there a problem associated with that? And the answer is yes, there is. So food waste is sort of the most labile component of solid waste. It's easy to degrade. Microbes immediately start degrading it when it gets into landfill. And in the process of the degradation process, water gets squeezed out of the food waste. And when the oxygen in the system is consumed, ultimately that results in fermentation, which produces a lot of organic acids. When the pH is depressed, that increases the solubility of metals in other components of the waste stream. And so ultimately that's going to leach a high concentration of heavy metals, organic matter, and so on into leachate, which ultimately can reach our groundwater and surface water systems if they're not completely captured on site and managed. The other major aspect of food waste and landfill is the release of methane gas. another product of anaerobic digestion of food waste. And the issue with methane is that it's a major greenhouse gas with many times higher greenhouse gas potential than carbon dioxide. So circular systems are a way to reduce those inputs and waste materials by recovering the nutrient components of the waste materials and using them to regenerate the soils and agricultural production. So on the right side of this slide, you can see on the top right, there's a composting system. So the food waste has been chopped up, mixed with some additional materials, and is placed in a pile to degrade aerobically in the presence of oxygen. And the output of this material of composting is a stable material that can add organic matter to the soil, recover the nitrogen and phosphorus so that new plants can grow with less inputs required from either mined or produced fertilizer material. So that's all good. On the bottom right, there's a picture of Exeter Agri-Energy. So these are the two things with the balloony looking things on top are anaerobic digesters where manure and food waste are combined to produce methane gas. So in addition to ending up with a material that contains nitrogen and phosphorus, this process also can generate electricity through burning methane gas that gets produced. So we have recovery of nutrients that can go back to the soils and the generation of electricity, which can offset some of the fuel inputs, the energy inputs into the agricultural system. So this is a desirable state to get to a circular food system. However, there's always a but. There's a few risks associated with this approach. The food waste recovery hierarchy set up by EPA puts source reduction, feeding hungry people and feeding animals above industrial uses like recovering energy and materials from the waste. It's higher than composting and it's higher than landfill and incineration. However, it's a lot easier to send all of your food waste to one place and aim a little bit lower on the hierarchy. So one risk to setting up systems that actually do recover the compost and the nutrients from the waste is that kind of competes with the higher level uses of food waste. But what I'm concentrating on in this talk is more the possibility for contamination in the system. So unintended or mistaken incorporation of trace contaminants that can potentially generate more risk in our food system and increase the level of contamination in our food. So the contamination can come at any stage of the food system, from inputs at the agricultural production level where pesticides, fertilizers, antibiotics can be added into the system on purpose. Pathogens can be introduced into the produce through management options. So if there are animals on the farm, the use of manure can transmit pathogens onto the produce as it, before it's actually taken off the farm. And trace contamination can enter into the system at this point through uptake in the soil, in irrigation water, and from aerial deposition onto the site where the food is being produced. During processing and packaging, additives may be incorporated into the food, which wouldn't normally be in that material, so that can add a bunch of chemicals into the system. Contact with packaging materials may also influence the amount of contamination associated with the food, and through handling and storage conditions, you can end up with an increase in the number of pathogens in the food. Distribution and retail add additional packaging. There's more potential for pathogens to be introduced, and use of cleaners and many other things could potentially add additional materials into the food and food waste. At the household level, household items or packaging of other sorts can wind up contaminating the waste stream. Pathogens may be introduced again through storage conditions or contact with other materials, and pet wastes may be included. And finally, at the waste management step, some of the contaminants that have been introduced may be degraded or removed physically from the system before it's actually composted or anaerobically digested, which can reduce the amount of contamination that would be associated with materials that go back to the agricultural system. However, some materials simply are unaffected by the treatment. So in order to measure contamination within the system, we collected samples from two different regulatory environments. One is mandated diversion, where people are expected and told that they need to separate food waste from the other waste materials that they're producing, and voluntary separation of food waste from other materials. Massachusetts and Vermont both have these progressive laws that require large generators to separate their food waste from other contaminants and components of their waste. And so those materials are diverted away from landfill. And in Maine, we don't have any laws mandating the separation of food waste. And so it's all voluntary or subscription service. In no case at the time when we were doing the sampling, our residential customers required to separate food waste. So all of the residential samples were voluntary. We looked at 6 different source types: grocery stores, restaurants, hospitals, schools, retirement communities, and residences. And we were looking for physical contaminants as well as biological and chemical contaminants. But we removed the physical contaminants before we processed the samples to be measured. So I'll start with the results of the physical contamination. We saw plenty of physical contamination, up to nearly 40% of the waste sample was physical contamination in one case, and that went down to basically clean samples. One sample was all just trash, basically, and so we didn't have anything left to process, so we actually ended up with only 71 samples out of our 72. The kinds of physical contaminants we found were plastics, packaging materials, papers, fruit stickers, coffee filters, and gloves. And interestingly, the regulatory environment did not affect contamination levels when we just looked at regulated versus unregulated. But when we looked a little bit closer at the influence of the source of the material, we found that there was some variability there. And residential waste had higher levels of physical contaminants in it than grocery stores did. In our case, we did have regulated and unregulated grocery samples. However, in Maine, all of the grocery samples were going to a place that had a depackager. So the grocery stores were not required to separate out their waste, their packaging and so on, from the food waste. So we kept that set of samples aside. And as you can see, the grocery samples had a low amount of contamination, and the residential samples were quite a bit higher. So that actually accounted for this difference in the amount of contamination in the system. We were expecting the exact opposite, that people who were forced to separate their food waste would have a higher amount of contamination than people who were doing it voluntarily. And so once we took out the samples, the grocery samples, which didn't have a comparator in the non-regulated samples, we found that there was no difference. So our first hypothesis that voluntary participation would improve the quality of the waste was proven false. Next, we looked at heavy metals, and we found that there were not a lot of heavy metals, and that is in line with what is seen in the literature. All of the samples were below the regulatory levels. Most of the toxic metals and any of the the metals that were related to electronics were below the limits of detection, and of the detectable metals that we found, zinc and copper, both were below the regulatory limit. So this was really not an issue of significance to us. When we get to the halogenated organics, the issue here is that these compounds have a tendency to bioaccumulate and increase in amounts as you move up the food chain. And that's a problem in a system where you're putting materials into the food chain. So what we did to assess this was to look at a bulk measurement called EOX, extractable organohalides, and with the goal of not prejudging what types of contaminants would be in the system. And this measurement has a relatively high detection limit. So I was sort of alarmed to see that 14% actually had positive values. And the limit of detection here stated by the lab where we had this done was 5 parts per million, which is a high level for halogenated organics. So we took, that said, the levels that we saw were less than the regulatory limit of a similar test that's done for sewage sludge in Europe. The AOX stands for adsorbable organohalides, and those, the concentrations that we had were below their limit for that measurement. We took about a third of the total number of samples we sent for PFAS analysis, and of those, over half came back with a positive concentration of PFAS. PFBA, butanoic acid, so it's one of the shorter chain PFAS compounds, was found in more than half. So anytime we had a detection, PFBA was present. And what was interesting is we never got a hit for PFOS or PFOA, which are commonly the things that are found at highest values. And PFBA is more easily taken up by plants. So it's interesting that that's the one that we actually found here. It's a replacement compound for PFOA, and it may be used in food packaging materials. So that could be the source of the PFBA contamination that we saw. Again, the levels here in the graph are in nanograms per gram, so parts per billion. So they're a little bit lower than a part per billion in all cases, but so the concentrations are relatively low, but this is still of concern because of its ability to potentially build up in the food chain. The last group of contaminants that I looked at were biological contaminants. We rarely detected pathogens in the samples, so that's testament to the safety of our food system in the US. We did high throughput sequencing to see if we could find pathogens that we weren't looking for, and we did find some genera that contain pathogens, but that aren't exclusively pathogenic bacteria. And the highest abundance that we found were the pink ones, which are Yersinia. Yersinia enterocollitica is a foodborne pathogen, but there are also many other species of Yersinia that are present in soil and food. So this was not out of line with what has been seen in other food samples. But maybe of more concern to me was the presence of antibiotic resistance genes. Almost all of our samples contained some antibiotic resistance genes, whether they were tetracycline or penicillin resistance genes. And the issue here is that antibiotic resistant genes are typically present on mobile genetic elements, which means that bacteria can trade them. They can transmit them from one bacterium to another, and even bacteria that are not related to each other can share these genes. And it's bad if bacteria obtain antibiotic resistance from their environment if these genes get into pathogenic bacteria and are then not susceptible to antibiotics that we would use to treat an infection. So that's the issue here. And antibiotic resistance has been increasing over time. Over the last few decades, the incidence of antibiotic resistant pathogens has increased over time, along with the use of antibiotics in agriculture. And so there's some research out there suggesting that increases in agricultural use of antibiotics results in these higher levels and the decreasing treatability of some pathogenic infections. So overall, our conclusions are that the main issues of concern to us are the halogenated organics, especially the PFAS. We had a high incidence of detectable PFAS in the foods wastes that we were looking at, and the elevated antibiotic resistance genes. The antibiotic resistance genes have a better chance of being destroyed during treatment. And so we think that it'll be necessary to go in and have a look at what happens to them during the treatment of food wastes. For processors, the issue is that these are not contaminants that they can readily see, and they're both very expensive to measure. And so it's unlikely that it's going to be easy to solve this issue without sort of incorporating some regulatory efforts to try to reduce their presence in the food system as a whole. So we need to make our food systems more circular, and I believe we can do that. And it can be done, and it can be done safely, as long as we keep mindful of the issues that are out there and keep a close eye on the system as a whole. Thanks very much for joining me today, and I look forward to the opportunity to talk to you and answer questions.